joining us. Thanks for joining us this morning, everyone. We've got some great presentations today, so we're really looking forward to some great content. My name is Dale Pavlovsky. I'm one of the marketing specialists within Merck. I'll be one of the voices that you'll hear today, more as like an adjudicator for question time and between talks. The other voice you'll hear is Bernice Tan. She's the Merck field application specialist for this gene editing portfolio. But the main and the key voices that you'll hear from today are our speakers, our three speakers, being Gurpreet Balrey, Marcus Keatinge, and Fabien Delerue. So without further ado, we've got a tight schedule, so I'll hand it over to Gurpreet to begin the talks.
Excellent, thanks Dale. Good morning all, and thank you for taking the time to attend the webinar. As Dale said, my name is Gurpreet Balrey, and I've led the development and market introduction of our SIG-RNA, cRNA, trace RNA system, and I've also worked with the wider team to develop and launch our synthetic single guide RNA. So just some housekeeping to start off with. So this webinar is being recorded. Please do type your questions into the Q&A panel of BlueJeans. In the end, please do tell us in the post-event survey how you enjoyed the talks. The questions will be answered periodically or at the end of each webinar topic, and the audience will be kept on mute during the webinar. In terms of the agenda today, I'll give you a brief introduction and touch on reasons that a lot of researchers have been switching to the RNP format of CRISPR-Cas9 for animal model creation, but also for cell line models, but obviously the focus today will be on animal models. I'll also be touching on the benefits of using synthetic guide RNA rather than in vitro transcribed single guide RNA. I'll be followed, as was mentioned, by two of the people that I've collaborated with personally on our synthetic RNA and who are very well regarded in their respective fields of research. Marcus Keatinge works with Zebrafish at Edinburgh University and has been using our cRNA trace RNA system for a while now. And Fabien Delerue from Macquarie University in your very own region works on various animal models, including mouse. He too has been getting some brilliant results using our single guide RNA this time. So they're going to be speaking to you after me. Before we begin, I just need to read out the following legal statement. So the life science business of Merck KGaA Darmstadt, Germany operates as MilliporeSigma in the US and Canada. Now, Merck has been heavily invested in gene editing reagents for almost 15 years. We began by supplying gene editing reagents for prokaryotic research back in 2005 with the Targetron system. 2 years later, the next big development was supplying zinc finger nucleases, or ZFNs, where we commercialized products and services under an exclusive license from Sangamo as the first commercial targeted gene editing reagent for mammalian use. Moving forward to 2012, and we filed our first patent on CRISPR technology, and quickly were the first to offer a commercial CRISPR plasmid by 2013. We continue our commitment to supplying cutting-edge gene editing reagents for model systems of any kind. Now, building on that first patent application in 2012, and showing that we're really at the forefront of this technology and working on the very cutting edge, we now actually have 28 CRISPR patents globally covering various aspects that you can see there. So those 15 years of knowledge, expertise, and research means that we have an unrivaled ability to help you with your gene editing needs and any support that you require. In terms of our CRISPR portfolio, whatever level of expertise you have and whatever application you're using these for, we have the broadest portfolio on the market. Starting from left to right at the top, we have plasmids, in vitro transcribed RNA, lentiparticles ready to transduce, and RNP, which is made up of synthetic guide RNA and recombinant Cas9 protein. If your focus is on screening instead, then once again we have a variety of arrayed and pooled options. For any information you need, please do get in touch with Bernice, who was mentioned at the start. She's your local specialist for these products, and I'll have her details at the end of my section. So when we talk about synthetic guide RNA, what we're talking about is what we refer to as SIG-RNA or synthetic guide RNA, which is the trade name for our synthetic guide RNA. We're referring to both the two-part cRNA trace RNA system, which has the two RNAs involved in CRISPR-based editing as two separate, easily orderable products, or the one-part single guide RNA system, which has these two RNAs that are joined together using a stem loop structure. Now, as you listen to the talk, you'll find that there is a place for both options, and we will continue to offer both to our customers. It's well known that using plasmids can cause unwanted random integration of plasmid DNA into the host system, as well as a certain amount of stress as a result of the transfection. Continuous production of the Cas9 protein, as happens due to the method of action of plasmid, also gives a higher chance of off-target cutting. In addition, you have to choose a promoter, buy an oligo, and clone it in, and then send this off for sequencing. What we wanted to do was create a system which would alleviate all these time-consuming and labor-intensive steps. By having the guide RNA synthetically made, we're able to do this, and by adding the recombinant protein, we are also able to make a system that has immediate activity and dissipates within 48 hours, essentially becoming fingerprint-free whilst reducing off-target effects. You essentially order an oligo which comes pre-QC'd, can be chemically modified, gets to you quickly at a decent price, and allows you to focus on other aspects of your research rather than wasting time making the tools rather than doing the science. There are obviously advantages over IVT RNA as well, with the guarantee on activity provided, stabilizing modification additions that you can't do with IVT, the QC, and the ability to make guides of any sequence that's not necessitated by having a G at the beginning, as is the case with many T7-based kits. Now, we have a lot of data on the effectiveness of both our two-part and single guide RNA for both cell line and animal work, but we have two great speakers who will be speaking about their work, so I'll focus instead on some work that another collaborator of ours at MRC Harwell in the UK has been doing, comparing their in-house in vitro transcribed RNA versus our synthetic single guide RNA. This was done by the Molecular and Cellular Biology group run by Lydia Taboul. That's also a very well-respected animal model group. I'm sharing these slides exactly as Matthew Mackenzie presented at our Bite Size Bio webinar that he did for us earlier this month, which is why you'll notice they're not in our Merck format. Let's start off with some background here. So the group would normally have made their own in vitro transcribed single guide RNA using a bought-in gene fragment as a template and using a T7 transcription kit. They would then run an aliquot of this on an agarose gel to check the yield, purify using a spin column-based RNA cleanup kit, rerun on agarose to check yield, and then nanodrop for quality. On the slide here, you see the results of the comparison of their in-house made in vitro transcribed RNA and our synthetic single guide RNA. You'll see that the costs for the IVT come mostly from the purchase of the DNA fragment and from the transcription and purification QCs, whereas the cost for the synthetic only came from the purchase of the guide. Cost for processing was minimal, as all they had to do was resuspend the guide RNA and then run it on a nanodrop to confirm quality. Cost, therefore, from their point of view was similar. The biggest saving they found was actually on the processing time. IVT would take one of them 5 hours over 2 days, whereas simply resuspending the guide and nanodropping it meant they were able to redivert resources elsewhere in the pipeline, such as in screening of the founders and the G1 animals, which can be one of the most time-consuming things for them. So moving on to the in vitro testing that they did on synthetic guides versus their in-house IVT guides, they used a workflow based on the guided sgRNA screening kit from Takara Bio to test the efficacy of the guides. One thing to mention is that in vitro cutting is obviously not a guarantee of in vivo efficacy, but they do still perform the assay as it will help them weed out any guides that they are sure will not cut and therefore not waste time and resources microinjecting embryos with them. A template containing the target site is combined with the RNP, and efficiency is measured by the intensity of the bands on the gel electrophoresis. Note that this is not a side-by-side comparison of IVT versus synthetic guides, but more of a snapshot of what they saw over a number of projects. By measuring the band intensity, you can see that you get a guide efficiency for IVT guides of about 30 to 40% overall. When you look at synthetic guide cutting efficiency, you can see that their guide cutting efficiency is much more like their control, i.e. sort of 80 to 90%. So their conclusion is that in vitro, the synthetic guides cut very, very well. But that's not obviously the important bit for them, right? So the in vitro results looked good, so they moved on to the in vivo work. Now, when they switched from in-house IVT single guide RNAs to chemically synthesized modified single guide RNAs, they immediately observed an improvement in desired mutation rates, as you can see on the right. They saw similar outcomes from 2 suppliers, so they know that the improvement is due to using the synthetic guides over the IVT. Again, this is not a side-by-side comparison of the same guides, as they could not justify the use of additional embryos. Instead, they just looked at efficiencies over a number of projects. Although the survival rates of embryos transferred to pups born, on the left, was high in projects using the IVT guides, this is most likely due to the lower desired mutation rate, resulting in more wild-type pups. So the result indicates an increase in desired mutation rate when using synthetic guides over IVT guides, with percentages being closer to, being closer to 60 to 65% rather than 10 to 15%. One thing they mentioned was that guide RNAs may not necessarily behave the same depending on the mode of injection or electroporation or the types of projects you're using them for. So this data that I've just shown you is representative of null projects performed using electroporation into zygotes. Here are my details and those of my equivalent for Asia Pacific region, Alina, but more importantly, here are Bernice's details. As I said, we have a lot of our own data and data obtained from collaborators. For any of this, or indeed any other inquiry on our products, please do get in touch with her. Now over to Marcus for his insights into zebrafish transgenics.
Thanks. Can everyone see my screen okay?
Perfect.
So I'd like to thank the organisers for inviting me to here to speak today. I'm Dr. Marcus Keatinge. I'm a postdoc at the University of Edinburgh, working within the Dementia Research Institute. And I'm going to talk about a project that I worked on before that, when I was in the group of Professor David Lyons and Professor Katharina Becker, where I was part of a wider group, the Zebrafish Inflammation and Neurodegeneration group. And today we'll talk about the project where we used Merck's highly active CRISPR screening to identify critical regulators of spinal cord repair in zebrafish. So, as we all know, CRISPR is really good at making knockout models. But the problem is that although now any zebrafish lab can make a loss of function mutant quite easily, this still takes quite a lot of time. So it takes about 8 to 10 months to establish a mutant colony for experimental study. So the initial CRISPR injection is obviously very straightforward and fast, but you need to grow these injected individuals for 3 months, identify founders, cross these out to generate your F1. If you don't have a mutant allele in both sexes, you will need to then propagate this allele, and only then you can start to do your experimental analysis. And obviously, if you work with zebrafish, you'll be crossing too many different transgenic lines, which is still laborious and costly. So you can spend the better part of a year generating a stable mutant line for something which may or may not have a phenotype of interest. So it'd be great if this could be done a lot faster. So fortunately, in zebrafish, there is a way of doing this. It's where you use highly active guide RNAs to generate a F0 null, which is known as a crispant. So this has been around for a few years now. Here's just an example. So on the left, you can see a transgenic GFP line with GFP neurons, and the authors injected a highly active guide RNA into this line against GFP, and it eradicates virtually all of the GFP signal. On the left, we have a wild-type zebrafish, which is highly pigmented, but if you inject a highly active guide RNA towards tyrosinase, this virtually eradicates all pigment. So this is really exciting because you can use guide RNAs to create essentially a mosaic null acutely, which is this crispant, and it allows you to do rapid loss of function genetic studies in a vertebrate model system essentially in days instead of months. So instead of spending 10 months making a stable mutant line, you can do this in a week. So it's very powerful. And several methods have been established using in vitro transcribed guides to generate these crispants. And the really nice thing about this is, apart from the fact that you can do these fast loss of function studies, is it's fairly easy to work with several at the same time. So instead of just looking at 1 or 2 genes in your experimental paradigm, you can start to look at tens of genes, which means you can really start to have a scalable system and do some really interesting genetic screening in a vertebrate model system. I'll talk about a screen I did where we looked at 30 different genes, but there are some publications where they look at over 100. And so this is all in vivo in the vertebrate zebrafish. However, there are some problems with the current systems. Using in vitro transcribed guides, you can get quite high toxicity, up to 20 to 30% can be deformed. This has implications for your false positive rate. Guide diversity can be too small or too large. With in vitro transcribed guides, you sometimes have to change some residues in the spacer region to get efficient transcription. But if you change these residues, although you get efficient transcription, this comes at the expense of CRISPR-Cas9 cutting. And importantly, in some of these methods, guide RNA activity is not determined despite the inherent variability in mutation rates between all guides. So within our group, we wanted to create a method that would be robust enough to knock out any novel gene to make a CRISPR-tin, but we also wanted it to be rapid and inexpensive. We wanted frameshift mutations to be maximized to reduce false positives, whilst we wanted to keep guide diversity to a minimum to reduce false positives. So this is why we switched to this synthetic CRRNA system, which is sold by Sigma and Merck. So we've been using these for about 3-4 years now. So with your in vitro transcribed guides, you have the PAM site, which obviously every CRISPR needs, but these 2 residues here in the spacer region, you would quite often have to change to a GG to get this optimal transcription with T7. However, this comes at the expense of CRISPR-Cas9 cutting. With this synthetic RNA oligo system, there is no requirement for this for optimal synthesis, so the spacer region will be exactly how you define it. So the only design requirement is the PAM site, and this allows you to really target anywhere you want and really opens up a lot of extra space for genomic attack. I would argue also that it is more cost-effective than generating your own in vitro transcribed guides when you add everything together, including your template, your transcription kit, and also your bench time as well, which is important. I mean, it all adds up, and we find that these commercially available synthetic guides just basically save you a lot of time and a lot of money. We always use Cas9 protein because it is highly active and has a low toxicity. And we always test these synthetic guide RNAs in vivo, just like we would with our in vitro transcribed guides, because we find even these have this inherent variability in vivo. So we always use restriction digest to test ours because it's cheap and quick, but you can really use any method you want as long as it is quantitative. So we always design ours to these restriction enzyme sites. Any enzyme site, I can stress, can be used as long as your recognition site covers the Cas9 cut site, which is here by these red triangles, which is 3 nucleotides down from the PAM site. But we favor these 3 enzymes in particular because they have quite large recognition sequences of mostly redundant sequence, so they're fairly resistant to SNPs. But also they have PAM sites in the correct orientation at either end, so you can target either strand of the DNA that you want. So here is an example of just a standard restriction enzyme site for one of these RNA synthetic oligos. So all of these wells are from an individual embryo prep. It's really important not to pull your DNA so you can ascertain what your injection technique is like. And like I said, we design all of our CRRNAs to restriction enzyme sites, and a reduction in cutting of the restriction enzyme shows that your CRRNA is working in vivo. So these 4 lanes here are the undigested PCR products. The second 4 lane are from uninjected controls, which have had the restriction enzyme added, and you get complete digestion. And the last 8 wells are injected individuals with restriction enzyme added to their PCR products. And you can see that they have this undigested band because the CRISPR has mutated the restriction enzyme site, so you know it's working. So just to give an idea of the different levels of activity you can get. So in the top gel here, we have a really great CRRNA. So this gives you a mutation rate of 100%, which is really what you want for the CRISPR generation. It's really reached mutation saturation. Every single locus for that CRISPR in every single cell of the zebrafish has been mutated. The middle gel shows around about 50%, which I wouldn't want to use for CRISPR generation, but it could be used for traditional stable mutant generation. And the final gel has quite a low percentage activity of around about 10%. So I wouldn't want to use this for anything, and I would just redesign and start again. But what was really exciting to us when we started using quite a lot of these, so this is another highly active CRRNA, is that these highly active CRRNAs, which produce a mutation rate of over 90%, which you really want for this robust CRISPR generation, is the fact that they actually were incredibly common compared to in vitro transcribed guides. And this is important because when you make a CRISPR, to do this, you really need genome editing events to be as high as possible. And when we looked at all the different CRRNAs that we made that we bought from Sigma, we tested over 350 in total, we found that a large proportion of them reach mutation saturation compared to in vitro transcribed guides. So when we compared this to in vitro transcribed guides in the literature, they had very few highly active guide RNAs, so maybe 1% in this study, 10% in this study, but we found just under half of our guides that we tested with this synthetic system were reaching mutation saturation. And on average, the mutation rate was higher at 70%. So we found these were highly superior compared to in vitro transcribed guides. We wanted to see, using this data, if actually we could skip this whole in vivo testing phase just to save time. So we fed all this data into ChopChop and looked at different parameters to predict in silico activity of these guides. However, we did find that although there was a correlation between in silico predicted activity and actual in vivo activity, this correlation is actually very weak. So we would actually advise against using these rule-based designs for these synthetic guides, as these strongly will restrict your target choice. And as I mentioned earlier, to make a CRISPR, you want genome editing events to be as high as possible, but also you want frame shifts to be as high as possible. This is because if you have an in-frame mutation, this can make a still viable protein that will function as normal. So just to illustrate, this is just two alleles that I made in my first postdoc at the University of Sheffield. So these were two mutations that were very similar in size, 50 base pair deletions in the same area of a gene. One was in-frame, one was out-of-frame, and the in-frame mutation was viable. However, the frame shift mutation was homozygous lethal by TAVE5, which shows that it's really important to maximize these frame shift mutations. And the nice thing about these in vitro transcribed guides, the synthetic guide system compared to the in vitro transcribed guide system, is that these highly active synthetic guides are so frequent, at just under half, it's possible, if you just test a bunch of them, which is what we generally do for each gene, it's very possible to identify a pair very rapidly for each gene. So if you use two, this will induce biallelic frame shifts in 80% of cells, which are pretty much guaranteed to be loss of function. Now, you can increase your CRRNA number to increase this biallelic frame shift rate, but it will do so with diminishing returns and increase your off-target effects at the same time. So we think injecting two is a nice trade-off between getting a high level of frame shifts in your cells, but reducing your off-target effects and false positives. Now, this is important because although it is very easy to make a CRISPR, you want to validate your data with a traditional stable mutant line, this will actually take quite a long time and a lot of money. So this is why we really want to minimize these false positives. So just to illustrate how this works at the bench, as I said earlier, we were interested in spinal cord regeneration in zebrafish. So we wanted to do a screen to find modulators of this. So using a publication that came out in Nature Comms by my partner on the project, Dr. Themis Saroukas, we identified 30 genes that we were interested in generating CRISPRs of. So we found their orthologs in an afternoon, all 30 of them. We then designed 4 synthetic oligos against restriction enzyme sites per gene for our initial activity screen. So that was 120 in total, and that took a day to do all the design. We then tested all of these in vivo, which took 3 to 4 weeks. So we always co-inject 4 guides per gene just to save time on this activity testing phase. And then we selected 2 highly active CRRNAs per gene for CRISPR generation. If we didn't find 2 highly active ones in our first round of testing, we would just redesign and reorder. We would generally find 2 in the next batch. We then spent a few weeks subjecting these 30 crispins to our experimental paradigm, which is this spinal cord lesion regeneration, and then spent validation, which took the largest amount of time. So we validated with traditional stable mutants, which took 9 months. So just to show you what we actually do to the fish, so zebrafish can regenerate their spinal cord, and we're interested in finding modulators of this. So we cut the spinal cord at 3 days, and then using a transgenic line, this is the nice thing about these CRRNAs. You just, for crispins, you just inject them into any line you want, and it makes this knockout acutely. Using our transgenic lines that label the spinal cord, we actually watch over 2 days how the spinal cord will bridge this lesion site and repair. You can see with this white arrow. I'll show that again. So this is the lesion, and then the white arrows will show you the axons and the neurons bridging this lesion site. So with our screen, we subjected 30 crispins of these genes. These were mostly cytokines. There was some genes involved in neuroinflammation and in neurodegeneration. And out of these 30 crispins, we found 10 of them showed defects in spinal cord regeneration. So we took the top 5 that we were most interested in, and we validated these with traditional loss of function stable mutants. And what was really interesting to us is 4 out of 5 of these validated quite nicely, but one of them, this gene CST7, did not. So although we got a nice phenotype with the crispin, this was not recapitulated with the traditional stable mutant, which shows that this technology is really great, but you always need to do validation because there are still off-target effects of some form. So the guides for this gene were predicted to have low off-target effects, but it would suggest that other things are going on, which would suggest that it is important to validate with stable mutants. So 2 of the genes that we were interested in, we delved into the biology a little bit more. These were TGFB1A and TGFB3. So we initially used the crispin strategy, and we looked at macrophage number, and we found that macrophages were unchanged at the lesion site in these crispins compared to controls. However, when we looked at neutrophil number, we found that there was a large increase in neutrophils at the lesion site in our TGFB1A crispins, and this was accompanied by an increase in IL-1 beta levels as well, which we've previously shown to negatively affect spinal cord regeneration. So obviously these were just crispins, so we wanted to validate this with our stable mutant. So we then looked at our TGFB1A traditional stable mutant, and we found indeed this also had an increase in neutrophil number at the lesion site, which validates our crispin work. And at the moment we are looking at IL-1 beta levels in the stable mutant as well to really finish that off. So just to conclude, synthetic CRRNAs are great. They are highly active in vivo and are very, very much more versatile for targeting. I think they're a great product. They've saved us a lot of time and a lot of money over the years. They've allowed us to do these screens we never really thought would be possible to do. But it's still important to determine your activity for all of your synthetic CRRNAs because their activity can vary. We advise against using in silico prediction for these, as we find it is a poor predictor of activity in vivo. So we normally test multiple CRRNAs per gene, determine their mutation rates, and then identify a pair for CRISPR analysis. So, like I said earlier, we normally test 4. If we don't find 2 highly active ones in that first 4, we then just order another 4, and that normally does the trick. But we also will always validate with stable mutants, as we've shown with this screen, you can still get false positive. I'd like to just thank my funders who funded the project, who are Biogen, my partner on the project, Dr. Emis Daroukas, and the rest of the team, and especially our supervisors at the time, Professor David Lyons and Professor Katerina Becker, everyone at Edinburgh and Sheffield. Here is my email. Feel free to contact me and ask me questions. This publication is currently under review, but the preprint is on bioRxiv if anyone wants to look at that. You just search for my name and you should be able to find it quite easily. Thank you.
Perfect. Thanks, Marcus. So before we get into Fabien's presentation, Fabien will start sharing his slides soon. We did have an offline question for yourself, Marcus, so I'm just going to read it out. I am now interested in knowing whether Cas9 protein really does have an edge over Cas9 mRNA when used for injecting gRNAs for CRISPRs. Do you have any experience on this?
Oh yeah, I've used both, and injecting RNA in itself is toxic to zebrafish embryos, which is why we always, as soon as it became commercially available, we started using the Cas9 protein. We found, when we did side-by-side comparisons, we found it worked a lot better. Because it's in glycerol, you just keep it in the freezer and it, lasts for a long time. We find when we used Cas9 mRNA, there was batch-to-batch variability, and it was very, very sensitive as well. So this is another reason why we stopped using the Cas9 mRNA. We just use protein now.
Okay, perfect. I think that answered that question well.
Cool.
All right, I'll give it another minute or a couple of seconds to see if anybody wants to post any questions. Can't see any popping up, so we'll start rolling over into Fabien's presentation. Fabien, if you want to go ahead.
Yeah, thank you, Darren. Can you hear me?
Loud and clear, and we can see your presentation well.
Perfect. Thank you very much, and great work, Marcus. This is great. We are trying as well to do a bit of work with Zebrafish, but this morning we'll essentially show what we've been doing in terms of generating genetically modified mice using single guide RNA. And before I do so, I'd just like to, as a matter of disclosure, inform everyone that neither my employer, Macquarie University, nor my company, Ideal Genetics, or myself have any financial or otherwise interest in this presentation. The only reason why I'm doing this presentation is because I've been invited. So thank you very much, G, for the invitation. And also because we've been using Merck guides for the last few years, and they essentially work really well in our hands. So all the results I'll be showing you in a minute have been achieved using these guides from Merck. Having said that, for those who aren't specifically familiar with how you actually generate transgenic mice or genetically modified mice, I'd just like to give you a bit of a background history. As you can see here, in the 70s, the first ever transgenic blastocysts have been achieved using viral delivery, and this is quite relevant at the end of this talk because although at that time the AAV technology wasn't up and running, it gave already an indication that using virus was quite a great technique for genetically modifying the mouse genome. After that, in the early 80s, microinjection took over and became and remain nowadays the main technique to generate genetically modified mice. And lately, pretty much as CRISPR came out, the Jackson lab showed that it was actually possible to generate these mice microinjection free, and that is using electroporation. And this is what we've been using for the last few years in our facility, and I'm going to show you how we've been doing that. But maybe before I go into the actual results, I'd just like to go back to the basics of how CRISPR actually works. You probably are very familiar with that. When you want to generate a knockout, you obviously complex the guide and the Cas9. And when Cas9 induced the double-strand break, then somehow it may overwhelm the cell repair mechanisms and doing so generates some indels being either small insertions or deletions. Likewise, if you co-deliver this Cas9 RNP with a donor template, which essentially has homologous regions on the side of the cut here, then by homologous direct repair, you can do precise and targeted insertions. So obviously, I suppose everyone is familiar with that. But what I would like to discuss further here is the efficiency of this homologous recombination depends on several parameters. And the most important one, and that's something that is often not discussed, is that the size of this transgene here increases, then the efficiency of the homologous recombination decreases. And that is obviously a problem when you want to do large knock-ins. The second criteria is that if you use single-stranded DNA as opposed to double-stranded DNA, like as illustrated here, typically a plasmid, then the efficiency is drastically improved. And finally, some other criteria, such as the nature or the format of the template that you co-deliver with the Cas9 RNPs, are of importance, and I'll show you an example on that in a minute. Now that I've seen, I'd just like to go back to the first ever papers on CRISPR in mice, actually 2 cell papers, that showed that it was obviously possible to do knock-ins with quite high efficiency, but also that it was possible to generate these knock-in mice by microinjection, co-delivering a donor plasmid. However, over the years, the transgenic cores around the world tried to reproduce such outcome, and the reproducibility of such results started to be questioned, and people tried to understand what actually was going on. And I show you here on the right the outcome of different techniques that have been essentially studied in terms of efficiency. And as you can see here, if you use a double-stranded plasmid, the homologous recombination level is actually really low in mouse embryo. However, if you use single-stranded DNA, then it is drastically increased in the magnitude of around 10%. So we and many other transgenic cores have 2 main challenges when we are trying to generate these mice. The first one is, as I just mentioned, the fact that the efficiency using double-stranded plasmid, the efficiency of homologous recombination is actually less than 1% by microinjection. And the second problem, which you don't have if you do cell culture, is illustrated here. And that is the fact that the mouse embryo are protected with this membrane here, which is called the zona pellucida. And this is quite a problem because for us, if we are to actually use electroporation, the plasmids do not penetrate via the zona pellucida. So for the last few years, we wanted to develop a method that would still be microinjection free, so essentially electroporation, but we were struggling in generating large knock-ins like any other transgenic core, but eventually overcame these limitations, and I'm going to show you how at the end of the presentation. But let's go back to the very beginning, which is how we generate knockout mice by electroporation. This is quite common. A lot of transgenic core can do that, and when you do that, it is not uncommon to reach up to 100% homozygous in the progeny. So really the efficiency of CRISPR in terms of generating mutation is unprecedented. And because we can achieve such a high efficiency, typically when we generate knockouts, we do not deliver only one guide, but we systematically co-deliver two guides, as illustrated here. And the rationale for that is because you can excise a piece of DNA of a predefined size. And when you do PCR genotyping down the track, it is much easier to actually identify your knockouts, both the homozygotes who display a lower bands and the heterozygotes with two different bands. So yes, the efficiency of the knockout can be up to 100%. Now, if you try to generate point mutations, so in that case, small modifications, by electroporation in mice, once again, and as always using single-stranded DNA, you can achieve, we achieve up to 50% homozygous in the progeny. So this is an example of one of the different point mutation mice that we generated lately. It's a single G2A substitution. And that is something that, once again, all the transgenic cores do routinely with high efficiency. But we wanted to try and push the CRISPR efficiency a bit further and try what people refer to as multiplexing. As you probably know, CRISPR has the ability to be mutagenic at different loci at the same time. And indeed, this case, the idea was to try to take this gene, which is involved in Alzheimer's disease, and try to reproduce by electroporation to generate as many as possible as the point mutations that are clinically relevant, that is, point mutations that are found in familial cases of Alzheimer's disease in this exon here. So we simply perform one electroporation session, but in this session, we co-delivered 14 guides and 14 single-stranded oligo, as illustrated here. Then we let the embryo develop and collected all the blastocysts and performed next-generation sequencing on the blastocyst. And as you can see here, out of the 14 attempted point mutation, only one failed to actually be generated. So that shows you the efficiency which you can actually generate point mutations in mouse embryo by electroporation. Now, once again, in terms of efficiency, what happens when you increase the size of the transgene? And in that case, the idea was to try to tag a gene of interest, and the tag was about 130 base pair. This was done within the context of the p38 MAP kinase signaling pathway, and what the PI was struggling at that time with was his main problem was that the antibodies are not specific enough to discriminate the 4 different isoforms here. So the idea was to tag one of the 4 different isoforms and see after that if by Western blot one could detect the endogenous expression of this isoform, and specifically and only this form. So we did attempt that. In blue here is the actual flag tag just before the stop codon, and we essentially managed to generate such a mass. mouse model, and as you can see, the PI was quite happy because he could detect his protein and the isoform of interest. But in terms of efficiency, I'd just like to show you what was the outcome of our session, because this is one of the rare examples where we did 2 different sessions in parallel for the same genetic modification. Here, what you can see is the progeny. So we generated 26 pups, out of which, as you can guess in green, 1 was properly targeted and carried the tag. The other 2 here did carry the tag. However, they either have only a partial insertion or some unwanted bystander mutation in the vicinity. So we did another session, as I mentioned, with for this project, and in that case we used chemically modified single-stranded DNA, as I mentioned at the beginning of this presentation, based on several papers that showed that these chemical modifications here could somehow improve the efficiency of homologous recombination. So we attempted that, and although it is only one example and I cannot draw conclusions out of this, one can still appreciate that in that case we generated less pups, had a higher success rate because we generated 2 founders with the correct insertion and another 3 with either the correct insertion and bystander mutation or partial insertions. So this is the sort of efficiency you can get once you increase the size of the transgene. We also perform floxed mice, that is the insertion of 2 LOXP sites around, in that case, the exon 4 of this gene of interest. That was done using 2 guides and 2 single-stranded oligo. And once again, in terms of efficiency, we were quite surprised because the efficiency was actually really high. Out of 22 pups, we got 7 that did carry either 1 or the 2 LOXP sites, and 3 of them showed the proper insertion of the 2 LOXP sites on the same allele. That's obviously very important. So once again, multiplexing yet a very high level of efficiency. Now, the main problem, as I mentioned, is doing large insertions. And for those who are familiar with mouse transgenesis, our first attempt of doing large knock-in microinjection free was to target this ROSA26 locus, which is a safe harbor. So I put the original paper at the ROSA26 here. And we attempted in that case a 2.4 kb insertions. But as I mentioned, the idea was still to try to do that by electroporation. And we managed to do it using a technique that has been developed really recently. I refer you to these 3 papers where the authors actually use AAV to deliver the template. And they found quite a high level of efficiency after studying the different serotype and identified which ones were actually very efficient. You can see that AAV6 was in this case quite efficient. So if you are to do this, the workflow is actually really simplified, and it is illustrated here. All you need to do is to take your embryos and infect them with the AAV particles. In that case, this was AAV1, but we did use AAV6. So you infect your embryos for a few hours, and after that, you wash them and then perform electroporation to bring the Cas9 RNPs inside the embryo and eventually reimplant and genotype the progeny as a traditional workflow. So we did attempt that, and really surprisingly, what we ended up with was doing a first screen using on transgene or transgene specific PCR. So as you can see, our primers sit on the transgene here, and the outcome of the PCR was this. So we were really, really surprised, and I'm still quite surprised by these results here, that showed that the majority of the mice did carry the transgene. But as you can appreciate, if you stop your analysis here, among all of these positive mice here, random integration can still have occurred. So to check that the integration is actually targeted, what you need to do is what's called junction PCR. And it is really critical in that case that your primers sit outside of the homologous recombination arms here. So one primer sits on the transgene and the other one on the endogenous locus there. And if you do that when we did that, for the 5' junction, this is the outcome. We realized that most embryos, most mice, actually had a correct insertion at the 5' end. Actually, out of the 19 positive that we had here, 18 were properly inserted. Only number 30 here did not integrate properly. So we obviously, as you can imagine, did the same on the other hand at the 3' junction. And this confirmed exactly once again our results. 18 out of 32 are actually properly targeted. Only this number 13, which was positive here, is actually not a proper targeted integration. So this was really surprising, and we since then have confirmed this over different loci. So I won't have time today to present on a project, but we reproduce such outcomes. So here, more than half the project was actually properly targeted. One could be random integration, although my understanding is that AAV cannot actually integrate, so I suppose it most likely is more of an episomal vector. And we did a bit further analysis for that to make sure that these knock-ins were actually knock-in. We selected 4 of them randomly and checked for transmission. And 2 of them had normal Mendelian transmission. One here did not transmit, although it had only 1 liter. So it's possibly a mosaic, as we sometimes see using CRISPR. But what was amazing is that this one here actually carried 100% transmission. So this one was actually homozygous knock-in for this 2.4 kb transgene, which is quite remarkable. So I'm going to finish up by showing you what we used to be in the early days of CRISPR, which was doing microinjection, using Cas9 mRNA, and synthesizing ourselves our single guide RNA. Then recently we switched to electroporation only, or almost exclusively. I'd say 99% of our projects are done by electroporation. We use Cas9 RNP, where we complex the protein with the guides that we purchase from Merck. And we also analyze our progeny mostly by NGS. And the main interest of that, if you compare year in, year out, is that in terms of output, we more than doubled our capacity to deliver a project. So really, in terms of workflow for us, buying the guides was part of a more general idea of streamlining our process to achieve a better output. So that's it for me for today, and I think there we will take few questions.
Hi, Fabien. Yeah, thanks for that. That was fantastic. I did see a question. So for those that can't see it, I'll read it out. I have a question regarding the use of fluorescently labeled sgRNA. Do you have any data regarding the transfection efficacy of labeled and unlabeled sgRNA, and is there any difference to be found?
Does the question relate to me or Marcus?
I believe it's to you. It was posted during your session.
Okay, so yes, we did try fluorophores. We did not specifically try the fluorescent single guide RNA, but to establish our electroporation parameters, we used first labeled dextran, and then after that we used EGFP mRNA. And we did set up and identify the correct parameters for our embryo, which delivered maximum efficiency, but also preserved the development of the embryo. So I won't be able to share this data. However, I can tell you that I did that over different species. So not only in mice, but also in large animals. And I advise anyone who wants to try that into their own capacity to actually do this pre-screen, because that allows you to find the right electroporation parameters and visualize the efficiency of the electroporations.
Okay, perfect. If anybody else has any questions that they want to ask while we've got Fabien and Marcus both still on the line. If not, Gurpreet, did you have any final remarks, or are we happy to close the session here?
Yeah, no, I mean, I'll just make a comment on that last question regarding the labeled single guide RNA. So from a synthesis point of view, it's quite difficult to fluorescently label a single guide RNA, because it's already quite long, and a lot of the synthesis platforms that are used, adding the fluorescence normally on the 5' end, even though we synthesize 3' to 5' is quite difficult. We do have a number of trace RNAs that have fluorescently labeled markers on them. That's a little bit easier to make. But honestly, you know, what Fabien was talking about, it's used really as a transfection control. So it's used to check how well your transfection is working. You can't really use it for fact sorting or whatever the case may be, because the RNA is so small, and not all of it is complex with the RNP, that you do have a lot of free RNA around. So you can't necessarily link the fluorescence with an event that you're looking for. So as a transfection control, it's great, and it does work quite well. For sorting, it doesn't really work. But again, just to finish off with, it's easier to do that with the trace RNA, which is only 69 bases long. It's much harder to do with a chemically modified synthetic single guide RNA.
Thanks, G, for your comments on that. I think Dale has dropped off. He texted me, so that's why I thought I'll close this session then. Special thanks to our speakers this morning, Marcus, Fabien, and Gurpreet. And thank you so much to our attendees. It's early in the morning here in Australia, but thanks for attendees from New Zealand as well. Thank you, everyone, and have a lovely day.
Thank you, Fabien. Thank you, Marcus.
Thank you.
Thanks. Bye.